So a lot of people use histomarks to help them identify the relevant transcription factors to figure out which genes are active or potentially active that can be turned on. There are other histomarks that are also important. So we mentioned H3K4 trimethylation. It marks the promoters and tell us in that cell which type of genes has the potential to be turned on or can be or are already on. Another mark, as we mentioned, is H3K36 trimethylation mark, which tells us the polymerase is elongating along this region and is marking kind of the gene body, especially the exons. And so this is a seminal paper from the Zhang Ren's group in 2009, where they use existing histone marks to look at novel genes in the embryonic stem cell. And so what they saw was if this region, you can see this region has a K4 trimethylation, and in this region, and then nearby there is K36 trimethylation. That tells us there is a gene in here because this is the beginning of the gene with K4, and this is the gene body of the gene, which is marked by K36 trimethylation, which means that this gene is going to the left direction, right? There's another example like this. The K4 trimethylation is marking the promoter, and the K36 trimethylation is marking the gene body. So we know there's another gene going in this direction. And interestingly, they see cases when there are regions, people didn't know there are genes in here. But interestingly, we see a very strong K4 trimethylation peak here, and also K36 trimethylation marks nearby. That indicate there's a gene previously unknown, which is going in this direction. And this probably is marking the potential exons on this gene, right? This is, and then they can use RNA-seq later on. Sorry, 2009, this is like before, or at least at early days of RNA sequencing. When you use microarrays, if you don't have probes, you just don't know this region has genes. But later on, if you use RNA-seq, you can see, indeed, there is a transcript in this location that people didn't know before. And you can use this approach to identify novel genes, especially long non-coding RNA genes in the genome by these molecular beacons, right? There are also genes that are marked by what people call bivalent domain. This is, again, a seminal study from Brad Bernstein's lab, but the figure is coming from a different paper. So we mentioned that in the embryonic stem cells, there are active marks, or at least poised to be activated genes. They are marked by H3K4 trimethylation. The promoters can also be marked, especially CpG promoters, can be marked by H3K27 trimethylation near the CpG regions if this gene needs to be silent. And so you would imagine a gene is either promising K4 trimethylation or not promising a K27 trimethylation. But interestingly, in embryonic stem cells, there are thousands of genes that are marked by both K4 trimethylation and K27 trimethylation near the promoter regions or the CpG rich regions in the gene. And when people look at where those genes are, turns out these are very, very important developmental genes, especially transcription factors that need to be turned on later on as the cell differentiate. So somehow the cell knows that these regions are important. They not only have an on switch, they also have an off switch. But just in case that the on and off is not tightly regulated, in order to ensure the on and off are tightly regulated in the embryonic stem cell, before the gene is even turned on, these regions have the on-off kind of a gate, both marked at the promoter region, just so that later on, when the gene needs to be turned on, they can be fine-tuned and really, at the right cell at the right time, and it's turned on at the right level. And so these bivalent gene turnout is again an important beacon to the cell for later differentiation. But human, as a scientist, we are using this to help us understand, these genes are potentially interesting because the cell is telling us that this gene needs both an on and an off gate just to ensure a tight regulation. And that can tell us which genes are more important. And And you can see in here, embryonic stem cells initially is bivalent. It has both K27 acetylation and K4 trimethylation on this nucleosome. But as the cell differentiate, if this factor become really on, what will happen is that eventually this gene will only have K4 trimethylation, K27 acetylation when its gene turn on, and the K27, the off gate or the K27 methylation mark will be erased. Then this gene will be really on. But if it's off, then the K4 trimethylation mark will be erased. The K27 trimethylation mark is on. Say differentiation, you only need to use this transcription factor once, and then later on it's off. Then they will lose their K4 trimethylation mark and keep the suppressive K27 methylation mark. And eventually they might even have DNA methylation to silence it for even longer. But then the poised ones means that maybe this factor is still useful later in the developmental process. And so they are still in the poised state with the bivalent still available on the cell, on the region. And so this is bivalent gene. We mentioned previously, H3K27 acetylation marks both the active promoter and active enhancer. And the enhancers, they really are enriched near the transcription factor binding. But if you look at across the genome, these are the regular enhancers. It might be bound by one transcription factor, and you can see the histone acetylation mark here. You can see here, this is where the transcription factor binds, and that nucleosome is actually being squeezed out. So on the histone acetylation location, there's like no nucleosome, but the two adjacent nucleosomes are better marked. And then nearby, the signal become weaker and weaker. So if you look at like a bigger peak, this is kind of a few hundred base pair of a peak, but on the nucleosome resolution, you can actually see this trough right in the middle. And this transcription factor binding site is also interacting with other factors such as BRD4, MED1, and P300 in this really sharp region. And you can see here, K4 monomethylation also kind of have a similar signal with K4, K27 acetylation, but there are even stronger adjacent noise in here. But interestingly, there are also regions in the genome which have not only strong binding, their K27 acetylation signals are much, much stronger, not in terms of height, but also in terms of the width of the peaks, very broad and high peaks. And so in 2014, Rick Young called those regions as super enhancers, which has very, very strong signals for both K27 acetylation, PO2, MED1, BRD4 signals. Out of these different ChIP-seq, K27 acetylation is the easiest to do. And so now people are using K27 acetylation ChIP-seq to help define those regions that have super strong K27 acetylation signals. Interestingly, the genes, a lot of these super enhancers actually overlap with the beginning of a gene. And the genes that are near these super enhancers turn to have very, very important functions. What Rick Young discovered through later studies was that a lot of the cells, the key, really, really important transcription factors are marked by these super enhancers, which means that their nearby regions have transcription factor binding that induce very, very strong K27 acetylation signals. And so these are called like super enhancer target genes. And these genes are very important for the cell lineage, also for disease, for different disease, especially in cancer. These regions have very, very strong K27 acetylation marks. And the very useful thing is that these signals are more informative than increased gene expression. For example, if you look at gene expression between breast cancer and normal breast epithelial cells, there are many genes that are upregulated. Estrogen receptor is by no means a very, very highly overexpressed genes because in general, transcription factors are not expressed at high levels. But interestingly, when you look at the epigenetics on the K27 acetylation signal, you see huge, K27 acetylation peaks near the estrogen receptor gene. And it's a really important sign that estrogen receptor is very important in driving breast cancer development. And so using this K27 acetylation signal, you can see not only we can identify where the enhancers are, what transcription factors might be important, you can also see which regions are these super enhancer regions that indicate that the nearby genes are very, very important, that might be important for differentiation, the cell lineage, and also the disease-dependent genes. It almost indicates that the cell knows these regions need to be highly expressed, and they are using the epigenetic marker. They are like really waving the flags for the transcription factors to bind there to activate the gene to ensure these important genes are highly expressed in the cell. And so these are super enhancer targets. And then in 2015, Wei Li's group also discovered something called super promoters. If you look at the transcription start site of many genes, most of these genes have a strong signal near the transcription start site. Of course, if you look at high resolution, right at the middle of the transcription factor, there might be a little nucleosome-free region because that nucleosome right at the transcription start site are being squeezed out by polymerase 2. And this is kind of a zoom out, 10 kb resolution. So overall, you only see a kind of an overall peak. But interestingly, so you can see most of these peaks are sharp. Interestingly, for some genes, the K4 trimethylation signals are very broad, like this. So, control genes or these, housekeeping or regular genes are like this. But then there are some genes that are very wide. And some of these are immune-related genes, and others are tumor suppressors, well-known tumor suppressor genes. And just by looking at this, they saw some interesting things. For example, when they look at many K4 trimethylation ChIP-seq data in the public, they can, this is looking at like close to 200 samples. They know these are normal and these are cancer samples that people profiled K4 trimethylations before. And they noticed that, this one gene in the normal samples, you see this gene having like a super promoter, potentially indicating that also, again, is another, flag waving to say that this, tumor suppressors are important. The gene needs to be expressed in order to suppress uncontrollable growth. And for some reason in cancers, the super promoter signals are lost. These become, regular, just a couple kb peaks now. And super promoter genes will have a lower expression. And if you lose a tumor suppressor, the cell actually will start uncontrollable growth and then develop cancer. And so they are using this approach again to help them identify more tumor suppressors in the genome and figure out which genes might be important, right? So if you want to look at more oncogenes that drive cancer development, you do a K27 acetylation ChIP-seq and look for those super enhancer target. But if you want to look for tumor suppressor functions, you do a K4 trimethylation profile, and you look at regions that are super promoters. And so I hope that you can see, depending on the experiment, depending on your biological question, you might do a different histomark ChIP-seq and then give you different insights. So human scientists are using the epigenetic beacons in the cell to also give us biological insights.